A warm welcome from me too. It's my great honor to introduce our first keynote speaker, who is Rebecca Fitzgerald, who is Professor of Cancer Prevention at the University of Cambridge and founding director of the Early Cancer Institute at the University of Cambridge. Rebecca originally trained in medicine in Cambridge and Stanford, and after some postdoctoral work moved in London, moved to Cambridge in 2004 to start a research group. Her research is really focused in understanding the evolution of cancer, not only from a scientific perspective, but really as a clinical problem, because the way I would paraphrase Rebecca's research, without stealing some of her thunder, is really to understand which people are going to develop cancer and how can cancer, in this case oesophageal cancer, can be prevented. Her research has won numerous prizes and accolades, which are too long to all be listed. But perhaps most importantly, her work was also awarded with an Order of the British Empire in 2022. With this, I'd like to hand over to Rebecca. I'm looking forward to your talk.
Thanks, Moritz, for your introduction. It's really great to be here. We've had a lot of fun organising this conference and trying to think of what topics we would cover. I wasn't supposed to be talking at all, actually, but we had to reorder things when one of the speakers couldn't make it. So here we are, but it's great for me to have a chance to share my science with you. So Moritz has paraphrased it very well. I'm very interested in one cancer in particular, which is cancer of the esophagus, which is a fantastic example of how cancers can evolve and develop through a change in the cell shape to metaplasia, as I'll show you, then dysplasia to cancer. And because it's a very long evolutionary process, we have great opportunity to intervene very early. So I'm a clinical gastroenterologist as well as a scientist, so I'm very much driven by what the clinical solutions might be. I will be talking briefly about cytosponge, capsule sponge that we've developed. So why am I interested in this particular cancer? So this is a really horrid cancer affecting your swallowing tube. And most patients present with very advanced disease. And the usual symptom they present with is difficulty swallowing and losing weight. And the treatment that we give patients, if we think that they have a chance at being cured of their disease, which is around 40% of the people that present to us, is to give them combinations of chemotherapy, often radiotherapy, and surgery to remove their esophagus. And even having gone through all of that, then the overall survival for all comers with this cancer is less than 20%. And that really hasn't changed over the last 30 years. So what could we do differently? So the idea is to think how we could be much more proactive and think about who the population is that it's at risk, and then to do something much more simple to identify the people who have a precancerous condition so that we can separate those at high risk from those at low risk, and then do something to prevent the cancer developing or to treat it very early on. And we know that for screening detected cancer, at the point at which we find something which we can treat down the endoscope, we can completely transform the outcomes. So rather than a less than 20% 5-year survival, we get an over 80% 5-year survival. So it's completely transformative. Not only is the survival much better, but this treatment is outpatient-based. So you can treat them, literally bring them in for a day session, go down the endoscope go down the esophagus with the endoscope and remove the early lesion, or burn away the rest of the Barrett's with ablation therapy. So early diagnosis is definitely key in this cancer. So what are we trying to diagnose early? So the pre-neoplastic condition is called Barrett's esophagus. So I'll just briefly explain to you exactly what this is, because this is kind of the basis for the rest of the talk. So the normal esophagus is lined by squamous epithelium, which is very like your skin. And at the point at which your stomach and esophagus joined, that squamous esophagus of the stomach changes to a columnar lining for basically the rest of the gut, right down through to the lower part of the rectum and the anus, which is then squamous lined again. So your gut is mainly columnar epithelium, apart from the squamous swallowing tube. And at that transition from the squamous lining to the columnar lining, at the junction between the esophagus and the stomach, that's where these tumours form. And so these are endoscopic pictures on the top and the pathological H&E images on the bottom. And you can see this very dramatic change in cell type that's happened between the squamous esophagus and this pre-cancerous condition called Barrett's. So this is a switch in the cell type, and one of the interesting things that I'm going to talk about is what is that switch? How does it happen? Where have these cells come from that are Barrett's esophagus? So you can see when you look down the endoscope that the Barrett's epithelium, this metaplastic change, looks like a dark red salmon pink epithelium. I don't think I've got a pointer. So this is the Barrett's lining here. And that's the normal stratified squamous epithelium. So Barrett's always starts, this is the junction between the esophagus and the stomach, the Barrett's always starts distally at this junction and then grows up. And it can be anything from 1 centimetre to even 20 centimetres in the extreme circumstances. So this is the H&E image. Now the pathologist, if they got this sample, they wouldn't know where it had come from necessarily. They might think that it was some small intestinal epithelium because these are villi and these are goblet cells. But this structure here is a submucosal esophageal gland duct. This is a native gland duct normally seen in the squamous esophagus from a duct that goes up here and then it sits down here. So the pathologist, if they happen to capture one of these submucosal gland ducts in the specimen, they would know that this biopsy has been taken from the esophagus and it's undergone a metaplastic conversion. And I'll come back to these little structures in a minute because one theory has been that perhaps Barrett's arises from this submucosal gland duct. And then in a proportion of patients, this Barrett's can gradually progress to dysplasia. And I don't know if you can see, but this lining here is starting to look a little bit irregular. So this is a dysplastic lesion, and under the light microscope, the cells are beginning to lose their polarity, have enlarged nuclei, and so on. And that can then progress to esophageal adenocarcinoma. So that's the linear sequence of events that we're kind of taught in medical school. And one question I've been asking is, how does this transition occur? What's the cell of origin? And is this process necessarily correct? Do you always go through these stages, or can you go directly from here to here, or even here to here? Do you have to go through dysplasia? And these questions are important because if we want to come up with a prevention strategy, we need to really understand what these events are so that we intervene in a precise and effective way. So in terms of the cell of origin, you might think that this squamous epithelium could transdifferentiate directly to this metaplasia. So transdifferentiation is when a mature differentiated cell type converts to another differentiated cell type. Or perhaps the Barrett's actually comes from the top of the stomach lining, the gastric cardia, because these are columnar cells here. So perhaps that's where the Barrett's come from. Or perhaps it comes from the submucosal gland ducts. These have all been theories. So why do I say, is Barrett's necessarily the obligate precursor? So as I just said on the previous slide, maybe you could go all the way and jump from the squamous epithelium straight to a Barrett's cancer. Do you have to go through this Barrett's precursor stage? And the reason I've been kind of rethinking this question recently is because most of our patients with esophageal cancer present de novo. We never knew they had Barrett's esophagus. They just present with their swallowing difficulty. And sometimes we can see the Barrett's, so we know it came from Barrett's. But very often, actually about half of the time, we can't see any Barrett's there. So was there Barrett's at all? Or has the Barrett's just got overgrown? And we've also done a study, and it was a back-to-back study with the Mayo Clinic. And we both found in our cohorts that these were large cohorts, that when the Barrett's is visible adjacent to the cancer, those patients do better than the patients when there's no Barrett's visible. So that might just be because these cancers are more advanced and they've overgrown the Barrett's, or possibly these are two different phenotypes, two different diseases come from a different starting point. So these are the questions that arise then that I'm going to go into. Where does the Barrett's columnar epithelium actually come from? What's the cell of origin? Is it the obligate precursor to esophageal cancer? Can we define the molecular genetics of the progression so that we understand the patients at low risk versus high risk? Because most people with Barrett's won't progress. And then, having got some understanding, how can we actually use this information in a practical, clinical way? So let's start then with the origin of Barrett's esophagus. So these are the theories which have been debated over many years, over decades, and there are multiple publications around each theory. And it's one of those things which is a hard question to answer, because we can't do the experiment in humans. We can't really, it's very difficult to prove. And the animal models are actually very poor for this disease. There's no very good animal model. So it does make it difficult to study in a dynamic way. So just to recap, the theories are transdifferentiation from esophageal squamous cells through to Barrett's metaplasia, expansion of the gastric cells, or regeneration from some residual embryonic cells has been suggested, or transitional basal cells. I'll come back to that in a moment, or regeneration from these submucosal gland ducts, because of course these are glandular structures that are buried deep beneath the squamous epithelium. So to tackle this question, as I say, it's difficult to prove because we can't really do the dynamic experiment in a human, and there aren't good animal models. So we took several different approaches to try and look at this. The submucosal gland actually had never been isolated before to characterize it. So seeing as that's one of the key hypotheses, we wanted to actually see if we could dissect out the human submucosal esophageal gland and characterize it. And so we were able to do that using whole mounts. And then we used a single-cell RNA sequencing approach to really characterize all the putative cells of origin and compare them to Barrett's esophagus, and later in the study to esophageal adenocarcinoma, as I'll come to. And then we performed some genetic lineage tracing based on the somatic mutations, knowing that these are not very abundant mutations because we're looking in normal tissues and pre-malignant tissues, but could that give us a clue? And then looking at the methylation profile and the open chromatin to as well do comparative studies. So this was the work of John Zhuang, postdoc in the lab, who did really beautiful characterization of these submucosal gland ducts, and was able to, I won't bore you with all the details, but was able to characterize in exquisite detail what the expression profile is of these structures, including the mucus acini through to the intercalated duct, the main duct, and then up to the squamous esophagus. Worth just pointing that keratin 7 is highly expressed in the duct cells. And that turns out to be important because two of the theories are around residual transitional basal cells or residual embryonic cells, and both of these papers point out that these cells have high levels of keratin 7. And we think that probably what they were profiling in these models, in human patients and their mouse model as well, was an esophageal submucosal gland duct, which harbors keratin 5, 7, and P63 positive cells. So to look at the similarity across the differentiation stages, and this was done by Carol Nowicki-Usuk, now at DKFZ, we basically took every single putative cell type that people have said might be the origin. So we used disease-free organ donors. So the esophagus and stomach aren't transplanted. So these were people who had sudden death and their organs were being transplanted. But this was basically surplus to requirements. So we took normal squamous esophagus. We took cells just straddling the junction between the squamous epithelium and the gastric epithelium, and cells from the top of the stomach. And then we took similar samples from Barrett's patients. So this is normal squamous esophagus, which is now going to be more proximal than the normal squamous of the donor, because we're going above the Barrett segment. And then we took samples again from the squamocolumnar junction, but now this is the junction not between normal stomach and normal esophagus, but between the Barrett's and the normal squamous esophagus within the Barrett's, and again the top of the stomach. And then we did the single-cell RNA sequencing, And then we were able to basically create the tree and look at the comparisons of these cell types across these different tissue types. And what you find is that all the squamous cells are very similar, not surprisingly. Interestingly, those are submucosal gland ducts, not sure I can see it here, are very similar to the keratin 7 high cells cluster with the squamous compartment, not with the glandular compartment. And then the columnar, Barrett's, and gastric cells all cluster together. The goblet cells, which are the most differentiated pathonomic cell of the Barrett's, are kind of separated on their own there. And then we've got a bunch of stromal and immune cells. So there's a lot of similarity between the Barrett's and the gastric cells. Now, of course, similarity isn't causation, but at least for the first time we could really compare in exquisite detail these different structures and see where the submucosal gland ducts lie. And then similar sort of approaches to compare the methylation profiles and the open chromatin, again comparing normal squamous esophagus, Barrett's esophagus, gastric cardia, and submucosal glands. And basically in all of these experiments we find similar things. So here the submucosal glands cluster with the squamous esophagus, and the Barrett's sits next to the gastric cardia. Barrett's does have specific methylation profiles, not surprising because you've got this mosaic of intestinal cell types, gastric cell types. So it's not exactly resembling the gastric, but it sits closer to this in the branch of the tree than to the squamous esophagus. What about the somatic mutations, to use that as a kind of lineage tracer? So here basically we're doing 60x whole genome sequencing, comparing normal esophagus Barrett's and normal gastric epithelium. And we want to see what mutations in the Barrett's, whether they are shared, whether we find any mutations, and these are very lowly abundant mutations in these normal tissues, but whether there's anything in the normal esophagus shared with the Barrett's or anything shared with the normal gastric epithelium. So we found cloning mutations in the Barrett's in 4 out of 5 cases, and they were always shared with the normal gastric cells, but never with the normal squamous. And here's one example. So this particular patient, these are the shared mutations with the gastric, nothing shared with the squamous. And we then confirm these mutations using Sanger sequencing that we'd seen in the whole genome. So from all this, we hypothesize that the normal gastric, and I haven't shown you the transcriptional data, but what we found from these studies was that the normal gastric differentiated cells are what we think is the cell of origin for the Barrett's esophagus, and that this occurs through transcriptional reprogramming through c-Myc and HNF4-alpha to give you the Barrett's metaplasia. So that's all well and good. We're saying that Barrett's, we think, comes from the gastric cardia, but it still doesn't tell us whether Barrett's is a necessary step for cancer to develop. As I said, often we don't see the Barrett's there when a cancer is diagnosed. And I was recently looking back at this paper, which was a paper which I remember reading it when I was on a plane on the way to a conference, actually, and realizing that this was a very important paper. So Jesper Lagergren is at the Karolinska. He's an epidemiologist and a surgeon. And he did this really painstaking study. Sweden have fantastic population records. So they were able to find all the people that had developed esophageal adenocarcinoma, and he did a nationwide population-based case control study involving personal interviews with patients and family members. It was actually a huge effort, and was able to show that reflux symptoms were a very strong predictor of esophageal cancer. So if you look at recurrent reflux symptoms versus those who were asymptomatic, didn't have any reflux, the odds ratio was 7.7. And if you looked at those with severe symptoms, so they were having frequent symptoms for over 20 years, then the adjusted odds ratio, this is really extraordinary, is 43.5. So this is a very major risk factor. This is for true esophageal cancer. When you take cancers just at the top of the stomach, the odds ratio goes down to 2. And what I noticed going back to this paper was that he also said the strength of the association with symptoms of reflux was virtually identical for patients with cancers who had Barrett's and those who didn't. Data not shown. I actually called Jesper up to see if he could dig out this data if he still had it, but sadly they don't have it anymore. But again, it just made me think, am I barking up the wrong tree here? Because all of our work, on our clinical work and the test we've been making that I'll show you to detect Barrett's, if Barrett's isn't the necessary precursor, then we're going to be not having any impact with our screening test. So to try and address this question, we've made use of a huge amount of data, which is a fantastic collaboration from across the UK. And this is a real shout out to the people that helped us do this, but also for any of you working on human cancers, having the cohorts is so, so important. If you're working on a common cancer, it's not so difficult, breast or colon, but if you're working on these less common cancers, then having the big cohorts and working with clinical collaborators and really carefully collecting the clinical associated data and then creating multi-omic data sets for us has been pivotal for the work that we've been able to do. And this is work done by Maria, who's in the audience, who's speaking later on. And she was helping supervise Sujath Abbas, PhD student. And one of the aspects that they were looking at is the mutational signatures or the mutational processes across the evolution of this cancer. So here we've got cross-sectional cohort of Barrett's, primary tumours, esophageal adenocarcinoma, and a rather smaller cohort of metastases. So it's difficult to make conclusions with this rather small cohort, but nice numbers of the Barrett's and the primary tumour. And then ascertaining the key mutational signatures, there are 14 in this cancer type, and then trying to understand when these signature processes are laid down and whether they are different processes at the different stages of the disease between Barrett's, primary tumour, and metastases. And these are the key mutational processes which seem to have some difference between these different stages of disease, and perhaps give us a clue about the early changes which are pivotal for this disease to take hold. So signature 17, A and B, these are the cosmic signatures, so T to C and T to G in the specific trinuclei context, is a really dominant signature in this disease, and you see it really, really early on. We do also see this base excision repair signature, and we can see it seems to be important, particularly in the primary tumour and less so in the metastases. And then apobec and aging, again, in the primary tumours. And there are also DNA damage repair signatures, which are important. But when we look at the abundance of these signatures, signature 17 absolutely stands out. So the size of these circles shows you the median exposure of these signature types. So there are two versions of signature 17, but they are very similar. And you can see that they really dominate in terms of the exposure. Signature 2, the apobec, and the SBS 30, which I just pointed out to you, they might be kind of subtle differences between the stages of the tumour, but you can see they're really not very highly prevalent. And here's the SBS 40 aging signature, which is fairly prevalent and increases as you'd expect across the disease stages. And this is looking at it in another way. And again, signature 17 really stands out as being the most important prevalent signature in this cancer type, and it's laid down really, really early in the Barrett's. And this includes non-dysplastic cases of Barrett's esophagus. So that gives us a clue, and I should say, we haven't really done the analysis in quite this way, but we see this very prevalent signature in all cancers, regardless of whether or not we can see Barrett's there. But can we look more specifically at this question of whether Barrett's is the precursor by taking all the cancers we've got in our cohort and dividing them up into whether or not Barrett's is identified as adjacent to the tumour. So for this, we took over 2,000 patients. This was quite detailed phenotyping. So we were looking at whether the endoscopist or the surgeon could see the Barrett's adjacent to the tumour, or if the pathologist could identify the Barrett's adjacent to the tumour. So if either the pathologist or the clinician could see Barrett's, we called these Barrett's positive esophageal adenocarcinomas. There were over 1,000 of those, and then there were 880 where no Barrett's was declared. And then later on in the analysis, we had some Barrett's-only samples as a comparison. And then we had collected a lot of epidemiological data, so we could basically compare across these two phenotypes for all of these clinico-demographic risk factor data. And then we had whole genome sequencing data on a good subset of them, so that we could look at the mutational loads, signatures, driver genes for the cancers with or without Barrett's esophagus. So that then when we integrated the data sets, the epidemiology and the whole genome sequencing, we could ask the question, do we think that all these cancers are the same? And they've probably arisen through the same pathway. So this is a very busy slide, and I couldn't fit it on one slide to show you the cancers with or without Barrett's. So these are the cancers without discernible Barrett's, and the next slide is with the discernible Barrett's. It's a very heterogeneous cancer. P53 is by far and away the most prevalent driver gene mutation, occurring in 73% of these cancers without discernible Barrett's. And then you have a smattering of other mutations. And as I'll show you, you have to take my word to it to some degree on the next slide, it's almost indistinguishable, the landscape of these tumours, whether or not Barrett's is present. When we look at the age, gender, cigarette smoking, BMI, and heartburn, they're also very similar. With some minor differences, which I'll show you. This cancer is very much predominant in men versus women. Males are in blue there. We know that these are the main risk factors: cigarette smoking, BMI, and heartburn. We see slightly more cigarette smokers in the cancers without Barrett's. They tend to be overweight and obese, and heartburn is very common but not ubiquitous. Oh, there's the one with Barrett's. So I think, as you can see, it's really incredibly similar. So we can't really distinguish these in terms of the clinical demographics or the driver gene landscape. And here are some of the phylogenetic trees. I've just picked out a couple of examples. So green are the patients with Barrett's associated with the tumour that we can see, and blue are the ones where we couldn't discern any Barrett's. This comparison set here, they've both got TP53 and ERBB2 amplification on the trunk. And in this set here, they've both got TP53 and CDKN2A on the trunk. And these are generally very common drivers in Barrett's. So although there are some differences in these trees, reflecting the heterogeneity of the disease, and you get longer trunks with more of these amplification events in the more advanced tumours, stage 3 and stage 4, the fact that you've got these commonalities in the trunks probably suggests a rather similar ancestry. So one thing I said to Sean when we were looking at this data set was, have we got any cases where we knew they had Barrett's and they were undergoing follow-up for their Barrett's esophagus and they then got a tumour and we couldn't see the Barrett's anymore? Because that would be really good kind of proof that probably it has come from Barrett's. And there were 214 cases in our 2,000 odd that had a history of Barrett's surveillance, and of those, 21 had no Barrett's discernible. And here are two examples. So this is a 60-year-old male ex-smoker who was having surveillance for his Barrett's, and sometimes this happens, unfortunately, these patients get lost to follow-up. And then he presented 7 years later with a stage 3 tumour, and at that point, it wasn't evidence of Barrett's anymore at endoscopy. And here's an 84-year-old female that we were again seeing very regularly, and then she turned up to my clinic a few months ago and somehow she'd been lost to follow-up. She was now 84, so she was rather philosophical about life, but it shouldn't happen ideally. But she again had this stage 3 tumour and it's not obvious that there is Barrett's anymore. So if I didn't know that, the person scoping her wouldn't have associated with a Barrett's tumour necessarily. So we think that these cancers probably do evolve through Barrett's, but they may become, when they're more advanced and larger, they may overgrow the Barrett's so you can't see it anymore. And indeed, if you put all these factors into a multivariable logistic regression, and here's the forest plot looking at the odds ratio. So to the left is the association with Barrett's, and to the right is the association of cancer without Barrett's. So you can see that most of these lie on 1. Looking at signature 17, ecDNA, driver genes, whole genome doubling. The things which are different is that the cancers without Barrett's are lying to the right, and these are more advanced tumour stage. And mutations per megabase, I only got sent this slide by Sean this morning, who'd just done this analysis, so this is very hot off the press. And I said to Sean, I phoned him up just before I came to the meeting, and I said, well, what about the mutations? You didn't put that on the plot before, so what's happening there? Can you adjust for stage? And then he just sent it to me before I got up to speak. And if you adjust this for stage, it falls exactly on 1. So basically, the earlier stage tumours are the more Barrett's associated ones, so they may have a low mutational burden. So, you know, we can't, this is something which is very difficult to prove, but it does seem to me, I feel fairly satisfied that generally we're going through the Barrett stage. And that means that if we have a screening program and we're doing more proactive case finding to identify the Barrett's, that should be a useful thing to do to try and intervene early and prevent these people from progressing. Back to the science paper from Carol and John. One thing we did as part of that paper was we took the single-cell RNA expression data, and we took all of those single-cell motifs distinguishing the different cell types, and we put them onto the, we didn't have single-cell, unfortunately, data for our cancers, but we had bulk RNA-seq. So we used music to try and understand what the differentiated versus undifferentiated markers are in the cancers with or without Barrett's. So as you can see here, these are the normal tissues. So if we look at the normalized expression of the genes marking undifferentiated markers, we can see that the cancers basically have a very similar expression profile, whether or not there's Barrett's associated. And being undifferentiated, they're rather similar to the Barrett's as well. And these are the differentiated markers, so this includes the goblet cells, enterochromatin cells, and so on. And here, because Barrett's is a more differentiated state than the cancers, so the Barrett's is sitting up here, the cancers are here, but again, whether or not Barrett's is visible, they look basically indistinguishable. So from an RNA expression profile, that would also go along with our theory that they've all come through Barrett's. So this is our summary then of what we think is happening, that gastric cells are the origin of the Barrett's through this transcriptional reprogramming, and that in a proportion of patients these progress to cancer through the Barrett's state. And this is a modelling study which was done by Kit Curtis, which would also suggest that Barrett's is the precursor to cancer. So this was a model that Kit developed mathematically, in which she's assuming it's a linear progression going from normal to Barrett's through to dysplasia to cancer. And if she then takes this model and extrapolates from the published rates of Barrett's to the expected number of esophageal adenocarcinomas and maps it with the actual SEER data, it exactly corresponds. So she's assumed a linear pathway, and it does seem to fit. So I think there are quite a few bits of evidence now coming together to suggest that it is the true precursor lesion. So how does the Barrett's progress to cancer? Not all patients progress. And when we see patients in the clinic, we're always keen to stress to them and reassure them that most of them won't develop cancer. But what is it about the ones that do progress? So this was work from 10 years ago now. Jamie Weaver, who's now in Manchester, has a PhD in the lab. And we were starting to do much more sequencing, and we said it would be great to develop a panel to identify the patients at risk of progression. And so we created this cohort, and we created a cohort of patients with never dysplastic Barrett's followed out to 10 years. So these were really stable people that never progressed, that had multiple biopsies taken. These are Barrett's patients who had progressed to high-grade dysplasia, and these are esophageal cancers. And what I thought we were going to see was that we would see a sort of stepwise increase in an accumulation of driver gene mutations as we go through the stages. And what we found instead was, well, we knew about P53 already. We didn't need to do this experiment to see that. But P53 does truly distinguish between high-grade dysplasia cancer from the non-dysplastic Barrett's. We rarely see P53 mutations in non-dysplastic Barrett's. And thereafter, we saw a huge tail of mutations, and we could have carried the graph on, which all were very low prevalence. But you see them across all of the stages of disease, even the never dysplastic Barrett's. And Jamie and Karen, the postdoc, I said, well, you must have contaminated or muddled up your samples. I don't think it's true, but it really was true. So you just get a lot of stochastic mutations very early on in the disease, in keeping with the signature 17. What does change, though, is the copy number profile. So if you compare the copy number profile with the pre-malignant stable Barrett's to cancer, you can see the very, very stark difference. So it's really a copy number driven disease. And if we try and look at the triggers and the timing for that Barrett's progression, so here we're using clonality as a marker about whether something's occurring early or late. So clonal is early, subclonal is late. These are patients with indolent Barrett's that doesn't progress. So this is a cohort, we've stacked all the data here together. These are benign patients compared to patients who do progress to cancer. And here we're looking at the Barrett samples ahead of progression. And what you see here, you can immediately see that in the cases that progress, you've got a far greater number of copy number events. Here's loss of P16, which we know occurs very early. But the trigger here is the loss of 17p, P53. So biallelic loss of P53 is what then triggers this whole very rapid smattering of copy number events across a whole number of loci, and his whole genome doubling down here, which is also seen fairly commonly. And some of these copy number changes can be really very high-level amplifications, and we now know that these are due to ecDNA. But we only really see the ecDNA events at the point at which we're getting high-grade dysplasia and into invasive disease. We don't see them earlier than that. So this was work done joint with Paul Michel and colleagues at Stanford, using the Seattle cohort and our Cambridge cohort. And we saw these ecDNA events generally only triggered by P53 loss. And we could see more than one ecDNA event often in the same case, with different genes incorporated into these extrachromosomal circles. And Alvin, who I just showed his picture on the last slide, he's been doing some more characterization of these, and he's been using a combination of short-read whole genome sequencing and then some Oxford Nanopore long-read sequencing, and also created these patient-derived organoid models so he could really try and understand exactly what was happening and some of these are very complex events. And he could do some de novo assembly using the long reads so that we could understand and reconstruct these amplicons. And there are indeed hotspots of amplification occurring with these ecDNAs and breakage fusions. The common ones are just shown down here. So more than 10% of recurrence for KRAS, ERBB2, and GATA4 included in these ecDNAs. And occasionally we can capture them in an organoid, which is quite useful to enable us to do some longitudinal studies. So given that copy number is such a good demarcation between benign Barrett's and dysplastic Barrett's, the question was whether we could use the copy number as a biomarker for progression. And ideally, you want to find abnormal copy number ahead of the patient progressing, so you've got an idea they're at high risk. And we wanted to do something that was clinically potentially applicable and affordable. So this is shallow whole genome, 0.4x, and this is working on archival formalin-fixed paraffin-embedded tissue, because we went back to our cohort and we found patients who up to 10, 15 years ahead of their progression, we had samples available for that sequencing. So we had 90 patients in this study. We included 876 samples with follow-up out to 10 years. So we took multiple biopsies per time point and multiple time points. And this in a sort of snapshot is what the data looked like. So the samples were taken from 45 progressors and 45 non-progressors. So the progressors all went from non-dysplastic Barrett's through to high-grade dysplasia and early cancer, and the non-progressors stayed non-dysplastic. These are little esophaguses, so we took multiple levels of their esophagus within the Barrett's and multiple time points. And this is a stacked a sort of mountain plot of the DNA copy number gains in blue and the losses in green. And you can see, I hope, that there's more going on in the progressors compared to the non-progressor samples. And these are little plots of each case. So space is shown on the y-axis, space meaning the different levels of their esophagus, and time on the x-axis. So each little plot is a given patient, just summarized. The red boxes are what are deemed high-risk copy number profiles by the algorithm that Sarah developed. So you can immediately see that there's a lot more red in the progressors than the non-progressors. That's the first thing. If you look at the time plots on the x-axis, you can see that for many of these patients, they're high risk right from the first endoscopy. So that's potentially useful as a biomarker. There is some heterogeneity across the segment, and there are some inexplicable patients, like this patient here looked really high risk but didn't have dysplasia or cancer, so far at least. But it looks fairly useful as a biomarker. So then when we look at the predictions, this is up to 96 months prior to them getting cancer. So this is 8 years ahead. And this is at the time at which they have their cancer. And this is looking at the percent predicted correctly by our algorithm based on the copy number. So you can see at the time of they're getting cancer, this is quite kind of reassuring. This is just a check. 93% had abnormal copy number predicted by our algorithm. But even 8 years ahead, 47% of the cohort, about half of them, looked like they were going to go on to progress. We could already see that there was instability there. And of course, the closer you get to the endpoint, the more likely we were to find it. So in a completely independent validation curve, the area under the curve for predicting progression was 0.8. So if we try and kind of put a lot of information just schematically about what the molecular drivers are distinguishing benign to malignant disease, which I haven't got time to discuss in great detail, but you know, the pathologists get this right most of the time. That's the other thing to say. So their phenotypic assessment of the dysplasia, low grade and high grade, is pretty good. And then P53 loss is critical event most of the time, and even if they haven't lost P53, they'll have the pathway will be disrupted through MDM2 or another mechanism most of the time. And then it triggers aneuploidy, and I haven't discussed clonal diversity today. Carlo and Trevor, I think, is coming, would be a much better place to discuss the clonal diversity. And then the ecDNA, and I haven't discussed SMAD4, and actually Alex is in the audience who did work on SMAD4 at the point of invasion. But these are later events as you go from pre-invasive to malignant disease. So just for the last few minutes, how can we put this, all this information and knowledge into a simple test to diagnose Barrett's esophagus early? Because endoscopy, this is an endoscope, is our gold standard diagnostic method, but we can't be everybody at risk, who are the people with heartburn, particularly older men. The male to female ratio is about 4 to 1. You can't be endoscoping every person or every male with a bit of heartburn. It would be infeasible. So how can we do something much simpler? So some of you may be familiar with this, but we developed a very simple, super simple test that you could do in a GP surgery. It would basically collect a lot of cells from your esophagus, and then you could do biomarkers to ascertain what's going on. And we wanted something that was super quick, easy, inexpensive, pretty operator independent, and you could link the cells collected to a molecular test. This is the latest iteration of the device. So you just pop the capsule on the back of the tongue, swallow it down with some water, goes to the top of the stomach. Over 7 minutes, the capsule dissolves and out pops the sponge. And you pull it out, collecting about 1 million cells on the device. And you then send it off to the lab for analysis. I always have several of these devices in my bag, so anyone wants to volunteer after the lecture, you're most welcome. That's just to check you're all awake. So get lots of cells on this device, and then we can send them to a lab and look for evidence of Barrett's esophagus. The challenge we have with this is quite similar to the cell-free tumor DNA kind of challenge, so it's a very low signal to noise. So we've got a pan-esophageal sample, so we've collected cells from the top of the stomach, which we use as a quality control, the whole length of the Barrett's, and the Barrett cells will be in the minority within that sample. One question you might ask is, do you get a representative sample of cells from this sponge? So normally you go down with the endoscope, you can see the Barrett's, you take your samples within the Barrett's, and we take multiple samples. So the Barrett's can be, sometimes you've got 10, 20 centimeters of Barrett's. You end up with a ton of samples that the pathologist then looks under the microscope at. With the sponge, we basically get one sample, but it's a pan-esophageal sweep. So we did an experiment just to literally, this isn't for clinical use, but the experiment was to see whether we do get a good representation of the different clonal architecture in our single sponge. So this was an experiment done by Karen, working with, it's a collaboration with Illumina actually. So we did whole genome sequencing and then constructed a targeted resequencing, deep sequencing panel. And that enabled us to identify over 1,000 variants and construct the clonal architecture from the Barrett's esophagus. And this is what the clonal architecture looked like from this particular Barrett segment. So you can see that there were 6 clones in this particular Barrett segment. And you can see that clone 2 and clone 6 had p53, and that's where the high-grade dysplasia developed later on in this patient. But what was cool is from our single pan-esophageal sample, if we applied the same targeted resequencing panel, we could exactly recapitulate the same clonal architecture. So of course, no test is perfect. You're not going to necessarily grab every cell. You may miss a clone, but you get a pretty good representative sweep of cells. So I'm not going to go through all the clinical stuff because I don't think that's so interesting to this audience, but we've done a huge amount of work over 20 years to try and see if this test is fit for purpose, starting with preclinical studies, sort of feasibility studies, which were exploratory to test our assays, and then a pilot study in a screening population, and then a case control study to look at the accuracy of the test we'd developed. And then we did a large randomized controlled trial of screening versus usual care, which was called BEST3. So that was over 13,000 patients. And what we wanted to do in this screening trial was to see if we offered this test to people with reflux and heartburn symptoms, would that pick up more Barrett's than the GP usual care? And by GP usual care, I mean the GP will give you pills to control your acid reflux, and they'll only refer you for an endoscopy if the GP feels it's necessary. And basically, we found 10 times more cases of Barrett's when we offered them the cytosponge compared to usual care. And that was when only about a quarter of people, 25%, took up the offer of the test. And it was an intention to treat analysis. So if we could get more people doing the test, there'd be an awful lot more Barrett's out there. So we found 10 times more, even with a quarter of people agreeing to the test. And so this test is now in guidelines, European guidelines and the American guidelines, as a case-finding method. So this is when someone comes to you with symptoms, you could offer this as a triage test to see if they've got Barrett's esophagus. And what we're also doing is seeing if we can use this test not only to diagnose the Barrett's, but monitor progression to cancer. Because currently the gold standard is an endoscopy and multiple biopsies, which are then tested by the pathologist. And if you've got a little patch of dysplasia up here, these biopsy protocols can miss the dysplasia. And that's very well, so this is a bit fuzzy, but we now know from this and other studies that you can miss neoplasia using this protocol. So we really need better methods of doing it. So we've been seeing if we could use the cytosponge as a sort of triage to help determine how frequently people should have endoscopy with this biopsy protocol, depending on their low, moderate, and high risk, and whether even this could be better than the current protocol. And so this is how far we've got to with this. This was work done by Nastasia, a clinical fellow, developing a risk algorithm to divide people into low, moderate, or high risk, and showing that you can really enrich the patients who have dysplasia, which is in red, in the high-risk group. And we missed less than 2% that were deemed low risk. So we've been getting real-world evidence for using this test for patients with reflux and patients having Barrett's esophagus. To date, over 20,000 have had this test in the UK. And this has been particularly useful during COVID pandemic when endoscopy was shut down. And to scale it, we've been coupling the pathology readout with machine learning, artificial intelligence algorithms. This is collaboration with Florian Markowitz's lab, so that the pathologist doesn't have to assess every case in detail. They can have a kind of semi-automated idea to tell the pathologist where to look and to speed up the process. And a recent collaboration with Microsoft has taken this to another level. I'm speeding up because I'm realizing I'm over time. So just to finish off with, I've shown you we've got quite a lot of evidence for this test, but really the ultimate impact of screening is to show that you can reduce cancer mortality. And that is rightly a very high bar. And when we went to talk to the National Screening Committee in the UK about whether we were ready to use our test as a screening test, they said you need to do a mortality endpoint trial. And there's been a huge amount of debate recently in the literature about whether you really need mortality endpoint trials or whether you could use surrogates. So if you're interested in this kind of space, for example, for the Gallery test, the Grail test, other multi-cancer early detection tests, the question is, if you're finding more cases or earlier stage disease, is that good enough to suggest clinical rollout, or do you actually need to show that you detect it early and reduce death from the cancer? Because it is possible that you detect things earlier, but you just know about it for longer. You don't actually stop them dying from the cancer. And this is a paper that's just come out in JAMA looking at all the randomized clinical trials of cancer screening and looking at cancer stage compared with mortality as endpoints. And basically, not every cancer is the same, but I think the conclusions from both of these is that we can't get away necessarily from doing mortality endpoint trials. But if you're doing a mortality endpoint trial, it's a bit of a take a deep breath because you need huge trials and long follow-up. So we're just embarking on this now, and we need to randomize 140,000 patients. We'll be following them up for 10 years. So by the time I retire, I'll know whether it was all worth doing or not. So I think that's pretty much all I had to say. So where does Barrett's epithelium come from? So I think it is gastric cardia cells with transcriptional reprogramming, but we still don't really know how that relates to reflux as the trigger. Is Barrett's the obligate precursor? I think generally speaking it probably is. We've got a lot of correlative data to show that now, but we can't absolutely prove it. Can we define the molecular genetics of progression? Well, I think we've got quite a good idea that it's P53 triggered, usually leading to genetic instability. But the question is whether we can really translate that into good clinical biomarkers to reduce the risk. And I think we've had a good progress towards developing better non-endoscopic early diagnosis tools. I think they're feasible and promising, but we still need to show the mortality benefit of an enriched population in screening to ultimately make a difference to patients. So sorry for going over a wee bit, and thank you very much.
And thanks, Rebecca, for a really fascinating opening keynote. I'm sure there are plenty of questions in the audience. And I would especially encourage questions also from trainees at this stage.
Thank you for this beautiful talk, Rebecca. So we've seen there's a lot of heterogeneity when it comes even to Barrett's progressors, right? There is heterogeneity within the Barrett's. And I guess also between patients that have Barrett's and will progress to cancer. Do we understand currently how much of this heterogeneity is biologically and clinically significant? Do you think at some point, I guess I'm talking both about genomic and transcriptional heterogeneity, and do you think at some point we'll be talking about Barrett's subtypes that are clinically relevant?
Subtypes?
Subtypes, yes.
I mean, most Barrett's doesn't progress, so
I guess I'm talking about
if we could have a molecular subtype that would tell us, that would be, is that what you kind of mean?
No, I was talking more about the progressors specifically, yes.
Yeah, and then within the progressors.
Yeah, within the progressors, yeah.
Yeah. And it's hugely heterogeneous. So as you know, the landscape, once you start plotting it out, I mean, signatures helps a bit. If you start collapsing things into processes, then you do see some commonalities. So I didn't present the transcriptional stuff today, and this was Alex's work, but when you look not only at the driver gene landscape, but then the RNA sequencing and start thinking about which pathways are dysregulated, then there definitely are, you know, common pathways disrupted by a number of different mechanisms. So I think that is a very useful way of thinking about it, particularly if you're thinking about therapies. Whether those approaches can also tell you about progression risk, I think we haven't really determined. But you're right, it's hugely heterogeneous, and this has made it very difficult for us because you need big scale. This is a relatively rare cancer, so you need thousands and thousands. I mean, we know this from the GWAS kind of studies. You have to get up to the 20,000 plus to really start, you know, seeing the signals that are important. So I think that's one of the problems, because the prevalence is often, you know, less than 10% of these individual changes. So I think we need to know more about the transcriptional. So a lot of the work has focused on the genetics, because that's often an easier place to start, right? But then you need to marry it up with the methylation profiles, the transcriptional profiles, and then looking spatially as well, which we're only just beginning to do. So I'm sure there's a lot more to learn.
Thank you.
Not yet.
Is there anything I should? Yes. Thanks for this beautiful talk. And as you know, I've worked on Barrett's with Trevor and Carlo, and we found that generally, when we found clonal events in Barrett's, progressors or non-progressors, it was on 9p. And you showed it as well as the earliest event in general. And I was wondering if you find this 9p event in the esophageal cancers in absence of Barrett's, which could indicate that, you know, they have gone through Barrett's, because it was really the one marker that we could find quite ubiquitous when we did
true.
much
We should
region.
put it on our summary forest plot. Yes.
So you found it
Yeah.
in most of the cases?
Yeah.
Okay. Then
It's a good point.
Yeah.
Okay, Carlo.
Beautiful work and really convincing and exciting. A lot of great progress in Barrett's. I'm wondering about these sort of, I guess it relates to the molecular subtypes and sort of alternative pathways. When we published our whole genome sequencing study, it seemed like around 10% of the progressors didn't have P53, but they had GATA4 alterations. Have you seen that, and do you have any insights about what might be going on there biologically?
Well, we haven't seen mutual exclusivity, I don't think, in ours. And yeah, Alex, I am right in the GATA4. And actually, we see GATA6 as more prevalent than GATA4, I think, in the latest analysis. But we don't see mutual exclusivity between GATA4 and P53. There wasn't much mutual exclusivity.
I don't know if it was mutually exclusive,
right, okay.
But the ones that didn't have P53 at least had GATA4. There might have been some that had both.
I mean, we definitely see MDM2, and the women in the cohort, the females, which is of course less prevalent, looked a little bit different, interestingly, and they were more often P53 wild type. So that's kind of interesting. Yeah. Did you want to say anything, Alex? And I realise Alex is one person whose picture somehow wasn't in my
That's okay, I'm only mildly offended.
Slides. I'm very sorry about that.
Yeah, we did see, I remember we did see strong mutual exclusivity between MDM2 and P53. Like I said, there weren't very many kind of patterns like that. A few between, like, you know, RTK, signaling pathway members, but the MDM2 and P53 were really striking. We have really good power because P53 is very common, but that's definitely an alternative pathway. We did see lots of GATA4. No statistical relationship between the prevalence of those two events, but could well be a role, of course.
Hi, wonderful talk. So my question is related to the copy number variations. When you looked at copy number variations in progressors and non-progressors, did you find any correlation between copy number variations specific to certain genes in early stages of cancer to the rate of cancer progression?
Yeah, not really. I mean, there are some, but actually the global genome-wide copy number metric was much more predictive and useful, and because of this heterogeneity that we're talking about, it gets harder when you drill into certain loci. Interestingly, when we've done a similar study more recently on squamous cell carcinoma, there we really find certain arms, chromosomal arms, that are very predictive, that we put into an algorithm.
Thank you.
I have the pleasure of channeling the online participants. Yeah. So there's two questions from online participants. Rina Kansal asks, have there been studies to show any correlations between lifestyle variables and transforming genetic mutations?
Again, it's a power thing. So we're just able to start doing that now that we've really got these, the epidemiology data cleaned. So we know that smoking is a risk factor for progression. BMI seems to be more of a risk, well, it's a risk factor for cancer, but also for Barrett's, and it's not clear whether that distinguishes progression. But I think there's more to do that. And actually, Sean, who did that study, we've now done a prospective questionnaire as well, so that we can get much more detailed, rather than just from the notes, information about people's lifestyle factors. But you need again, because people reporting on lifestyle is quite difficult, and their diet and those things that might be important, so you need big numbers.
Yeah, and I think the question in particular was asking if there's a relationship with lifestyle factors and particular genetic transforming mutations, like maybe P53.
So it's just emerging. I mean, for example, the KRAS amplification rates seem to be more associated with smoking, and fewer of those have the Barrett's association, but that may be more advanced tumours. So it's confounded by stage, so you have to be careful. But I think we'll be able to start drilling into those. But you have to be very careful not to jump to conclusions, I think, unless you have huge numbers and very strong data.
Then there's a second question. William Dallywater asks, so Barrett's arising esophageal adenocarcinoma can be thought of as gastric cardia adenocarcinoma, but esophageal adenocarcinoma rarely takes on the morphology of true gastric arising cancers, which are a mix of adenoma-like and diffuse type. Is it interesting to explore why gastric cardia versus cancers arising from other gastric sites act and evolve differently?
So we didn't show this, but we published a paper in Cancer Discovery last year, I think, comparing gastric IM with Barrett's IM, and it's almost indistinguishable. And that was a kind of multi-omic study in single-cell sequencing. The diffuse type gastric cancer is a completely different entity. So the esophageal adenocarcinomas, even though we think gastric cardia is the precursor, they're a bit different in that these are true esophageal. Some of them, often go towards the junction, but these are different from the gastric cancers, even though the IM is similar. This is probably a longer conversation. But yeah, there are subtypes of these tumours in the stomach and the esophagus.
Splendid. We are exactly on time, so I would really like to thank Rebecca again for a stunning opening.